Welcome to this recording of the 8th BioExcel webinar. In this webinar, Stian Zoland Reyes from the BioExcel project will give a brief introduction to the Workflows and Pipelines interest group, and then our main speaker for today, Ravi Meduri from Argonne, will describe his work on large-scale analytical workflows on the cloud using Galaxy and Globus.
Okay, so I'll introduce the Workflows and Pipelines interest group. Now I added pipelines in there just to not confuse with workflows for moving biosamples around and things like that. This is a BioExcel interest group for users of scientific workflow systems using data analysis and pipelining, command line tools and databases, doing biomolecular simulation and modeling, and other kind of related software. And the workflow system we're thinking about is Galaxy, Taverna, 9, COMS, and also common workflow language. But there's not like a lockdown list of workflow system we're talking about. We want to look at as this interest group as a way for people to get together and figure out which workflow system can they use, what is good and bad about them. More like practical things, how you actually connect different kind of tools, how do you deploy this, particularly on HPC and cloud infrastructure, which is what we want to cover in BioExcel, and also around using Docker for packaging command line codes, so you can get the software installed without too much hassle. What can we discuss in the Workflows interest group? Well, kind of the floor is open, but here's some suggestions for things. We can, as I said, compare workflow systems. You can look about integration things, particularly with job management systems and things like that. There are different ways we can do this. There's, as I mentioned, we can do face-to-face. BioExcel has funds that we can do actual meetup with real people. We agree on a place and we fly in there, and we can provide support for venue, lunch, maybe a bit of travel. So that option is in there. It's worth to keep in mind. But of course, we can do it all electronically. The main point is the ask.bioexcel.eu. Now we have our workflows group in there, which I think we can see in here, where you can ask. This is not from the workflows group. This is from the Haddock group, which has already started. But this basically is a Stack Overflow style thing where you can ask a question and then get answers. You can ask to be notified by email or just follow it on the web. So we invite you all to sign up to ask.bioexcel.eu to discuss workflows. We also have an online chat on using Gitter. That's a web-based chat which have already been successfully used by other community, like the common workflow language people here using it. As you can see, you can be quite techy if you want to. You can just paste in code samples and things like that. I think this could be more like a kind of direct way of communicating when you're kind of debugging something or trying to figure something out together. You can also share workflows. My group in University of Manchester, we have the My Experiment website where you can share workflows. There's a group in there for sharing BioExcel workflows. There's of course just a mailing list. If you sign up to an interest group, you will be on the newsletter where we, not even once a month or so, we'll send you links to the different workflows. But it's up to you to decide. So I think we can follow this up on the Ask BioExcel website to discuss how you can interact with it. And this webinar forms the launch of the Workflows interest group. So don't be shy just because there haven't been much activity yet. It's because this is the very beginning of that interest group. You could also talk about BioExcel services, how do we fit in there, what kind of services we can provide. This is the webinar we're in now, and you can join the workflow interest group by going to the BioExcel website /interest group, and there's a tick box in there. At least tick workflows and pipelines. You can take some of the others if you want as well, and then you will be subscribed to the interest group. So I think that's it about the interscript, and we can move on to today's webinar, which is sorry. So today's presenter is Ravi Maduri, which I've known for many years, probably 10 years now. He's working at the Argonne Labs and University of Chicago. He's been working with workflows and reproducible science, particularly using large-scale computation. And today he's going to talk to us about Globus Genomics and how they are using large-scale cloud infrastructures to perform genomics analysis. So I think I'll give the floor to Ravi now.
Thank you, Stian. So let me try to share my slides real quick. Can you make me? Okay, I can share my screen. So thank you for this opportunity. Really appreciate it. I've been working with Carol's group and a few others in the European communities for a while. And quite honored to be invited to present some of the work that we've been doing. So as Stian mentioned, my name is Ravi Maduri. I'm a senior fellow at the University of Chicago and a scientist at Argonne. And we, so I thought it might be useful to kind of introduce us in a so that if folks are on the call would understand where we are and what we do as a group. If you haven't heard of us, so we're part of the Globus group. Globus is a software as a service capabilities developed and operated at the University of Chicago and Argonne National Laboratories. We are the group that I work in is spread across the two institutions under Computation Institute. We are a nonprofit organization because we work under the university, and our goal is to build solutions for nonprofit researchers. We have in the past developed a lot of capabilities, a lot of middleware software that was used for distributed computing in high performance data management and HPC. Our goal always has been to create software that would accelerate the discovery process, the scientific discovery process. In the course of the last 5-6 years, we have realized that one of the harder things to do with software for end users is to download it, install it, and configure it, and take advantage of all the power that is coded in the software. So we decided to take a sort of an easier route to make the software available, and we leverage software as a service as a way of disseminating the capabilities. So as part of the goal is to lower the barrier to entry for end users to take advantage of software and be productive and in process accelerate the discovery process. And this is as part of this goal, we created capabilities, services that I'll talk about today. Globus Genomics being one of them, and I'll also talk about some of the joint work that we've been doing with Stian and others in Carol's group for reproducible research under research objects and workflow sharing concepts. So I'm on full screen, so I can see the chat window. So if you have questions or if you would like to ask anything right away, please let the organizers know, and they will definitely let me know that there's a question. So let me quickly set up the problem space. One of the motivating factors for us to build Globus Genomics and operate it as service has been just the acknowledgement and understanding of some of the challenges that researchers were facing when doing large-scale analysis. And with Stian and Carol, we worked very closely with the National Cancer Institute here in the U.S. To enable researchers to do large-scale data analysis. And one of the so we've noticed that researchers often have two sets of challenges. One set of challenges is in the area of data movement and access, which we broadly classify under data management, and another set of challenges is in the analysis side. So if you look at the challenges in data management, especially in genomics and other scientific domains, data that researchers are using or want to use is distributed geographically. And typically the data sets are transferred to where they could be analyzed using FTP or SCP, and one of these protocols that are designed for an internet that had a very high latency and low bandwidth. So they are very suboptimal in terms of moving large-scale data sets. Especially in the U.S., I'm not sure if this is the case in Europe. If researchers are doing sequencing data analysis, they typically send their tissue biospecimen to a sequencing center to have it sequenced, and the sequencing center does the sequencing and copies all the raw data into a hard drive and puts the hard drive in a FedEx truck and ships it to the researcher. While this is, you could think this could be a reliable way of moving data. It's actually not. Our team has worked with various researchers who are getting sequencing done in China in BGI. And BGI also ships hard drives, and these hard drives typically get stuck in export control under in the border, or they get corrupted while in transit. So the challenge with researchers having to go through this pain is that they're essentially spending a lot of time either waiting for data or waiting to access the data, or waiting for the data to be transferred successfully. So as you can see, there's a lot of sort of waiting around and just babysitting the transfers so that they can finish successfully. And even when the data is ready, you want to move it to where you could compute on. Again, the processes that are used to move the data to where it could be computed on are also reliant on these protocols that are probably not very well suited to move big data sets around. And when you look at the data analysis side, I've taken an example of a genomics pipeline called the GATK best practices pipeline, which is very commonly used across a lot of genomics researchers. Typically, the norm is the shell scripting, and this is sort of it's done by people who are computationally more sophisticated, and while who don't want to kind of do repeated tasks on their command line, they typically resort to using shell scripts. So if you look at what goes on in these shell scripts, again, you move data to the compute node, install all the dependencies, like BWA. If you're doing genomics analysis, you figure out what are the different versions of the underlying Python that are R or one of the systems packages, and then write these shell scripts to sequentially execute the tools. And whenever I come across people writing shell scripts, something that Carol Goebel used to mention, shell scripts are like toothbrushes. It's very hard to share toothbrushes. It's very hard to share. It's not advisable to share shell scripts either, too. So these are the challenge with these scripts is that they sort of captivate the knowledge, and the knowledge that is embedded in a shell script gets it's very hard to transfer it to other people in the group. and they're very error prone, and they're very fickle. Depending on the day of the week, a shell script may decide not to work. I've seen these things happen. So one of the challenges, one of the anecdotes that I like to give here, we worked with a researcher here in the U.S. In the University of Washington. He's a very well-known researcher who works in the in neuroinformatics or neurodegenerative disorders like autism. His main area of focus is to work in kids who have abnormal brains and understand the relation between the size of the brain to the disorder. He had a fairly big group, a few postdocs and a bunch of graduate students, and two of his postdocs, because they were exceptionally good, they had offers from to become faculty members, and once they left the group, our collaborators' research came to a standstill because he couldn't do any analysis anymore because the postdocs had all the knowledge and all the shell scripts. So it's you know, the challenge here is that you have to figure out a systematic way of capturing knowledge, and in a way that this knowledge could be easily transferred across members of the group and across the scientific community, not only for continuity of your research, but also for reproducibility and validation of your results. So we've seen all of these challenges, and in addition to that, one of the some of the additional challenges that that have been more predominant with the advent of more and more research data being termed as big data is the challenges in rapidly validating a hypothesis, because now you have to sift through a whole bunch of data in order to do that, and scaling up the analysis after validation, and then once you validated and scaled it up, you want to apply the same technique to other data sets of interest. And I'll talk a little bit about this so that it gives you a bit more context as to where this is coming from. And then do all of this in a way it's reproducible, so that when you publish this research, somebody can not only just validate it, but build on top of it with confidence that you have used the right parameters or the right ways of analyzing this data, and to take the data sets and publish so somebody else can discover them. So these are some of the additional challenges that that researchers that we worked with and that we are collaborating with are facing. So Globus Genomics and Globus, the suite of services are built to address these challenges. So Globus Genomics is essentially a software as a service that runs on Amazon Cloud, that integrates Globus data management capabilities and identity management capabilities with the Galaxy, which is a popular workflow management tool that is developed at Hopkins and Emory and Penn State. So the idea for us, we've been working with the Galaxy community for a while, and if you look at the mailing list or researchers trying to use Galaxy, they have one or two options. One option is they go to public Galaxy and kick the tires, if you may, or try to use it in at a smaller scale, or the scale that is allowed by the public Galaxy. And if they have to use Galaxy at their institution, they have to go and hire a couple people, install Galaxy and set up a cluster and use it. Again, so none of this is impossible. In fact, a lot of people do it. But as you can see, this is the same problem that we had when we were developing Globus, where every time somebody has to install or take advantage of Globus software, they had to hire a couple people to configure the software to use it. So we thought, this is not a useful way of spending resources. So we thought we would create a capability, run it on cloud, as a service, so researchers can take advantage of Globus data management to move large amounts of data into Galaxy, and we created a set of 20 best practices pipelines on Amazon using the tools in genomics, so that researchers can start from one of these workflows instead of starting from scratch. And some of the additional capabilities that we made available is that, and I'll talk about this a little bit more, is making these workflows run at scale, and be able to match some of the resources that are available in the cloud automatically. So we have innovated on scheduling workflows efficiently with sort of economics of the workflows built into it, so that because you're using a public cloud, you want to minimize the amount of money you would spend on compute. So we leveraged spot instances on Amazon. So Globus Genomics, essentially, when a researcher runs a workflow, it sets up a cluster on Amazon using spot instances, runs the analysis at scale, and once the analysis is done, the cluster is dissolved, and the resources are resubmitted to Amazon, so you don't incur additional charges. So coupled with Globus, because Globus provides capabilities for integrating identities. So if you you could log into Globus using your campus or university credentials. You could manage data. Globus provides a high-performance data management services, so that you're not spending a lot of time babysitting transfers. It's fault-tolerant. By that, I mean most of the network transfers work better by a simple retry, because a lot of network conditions are transient in nature, so that when a transfer fails, you just if you could just retry it, it would work. So Globus automatically retries it for you and sends you a notification once the transfer is done successfully. So Globus Genomics is a service that runs on Amazon Cloud, leverages some of the spot instances capabilities, and integrates a lot of public data sets and a lot of reference genomes that researchers use, and so that a lot of time is spent on validating a hypothesis or running an analysis versus figuring out how to move data or how to run the analysis. So our hope is that with this capability, the productivity of researchers would go in the right direction. So some of the high-level technologies and services that we used to build this capability is the file systems on Amazon for Scratch and semi-permanent storage. So we use EBS and S3. We use a bunch of services for compute. We recently started using the Docker containers to schedule container services, VPCs for security and virtual private cloud networks, elastic load balancing for balancing depending on the traffic. We used ELB to load balance on the web server. Condor, HT Condor for scheduling these jobs, and the transfer identity management, we leverage Globus capability. All of this software is encoded in a bunch of Chef recipes. Chef is a tool that we used. There are a lot of other tools for configuration management. We use Chef for various reasons. So we have recipes that would allow researchers to that allows us to reliably launch these capabilities on Amazon, and we have a bunch of other services for monitoring and audit management, auditing capabilities, and RDS for databases and managing state using these databases, and identity and access management to control which account in Amazon that we use to launch these spot instances, and also manage billing using that. So these are some of the high-level services that we have used, and some of the capabilities that we built in the platform As this is a service, we manage it professionally, we support it, and we have, as I mentioned before, a whole set of best practices pipelines. So researchers don't have to start from scratch. They have a pipeline that they could tinker with, or they could change the parameters. Galaxy allows you to do this very efficiently, and we have added a whole suite of tools. We have over 1,500 analysis tools in Amazon. So one of the things that we've done with this 1,500 tools is that we have computationally optimized them. We generated profiles that will allow us to understand what is the right instance type on Amazon that makes this tool work without trailing. So we have those profiles so that we can schedule the analysis cluster required to run this analysis on various Amazon resources. And we do, as Globus, using Globus, we support reliable data transfer into Globus Genomics from various sequencing centers, and all of this is available as a subscription-based service, so that our goal is to achieve sustainability with this service by providing a good service, and there's a subscription associated with the capability, so that the subscription would allow us to manage the service, make it better, and just maintain it and support the people that maintain it. So let me quickly talk about some of the capabilities that we developed in order for this service to scale. The tool profiling service is something that I talked about. This essentially is a capability that we run on Amazon constantly. When we add a new tool, the tool gets picked up by the profiling service, and it does run this tool on various compute nodes to understand the CPU, I/O, and memory attributes of the application. Creates a profile, puts a profile in a database. So when a workflow attempts to run this tool, it consults the profiling service, and the service, the profiling service, would let the scheduler know that this tool requires a higher, a kind of an instance type, and then that instance type is transferred to the provisioning service that we have, which looks at what are the available instance types and what they cost on Amazon spot market, and essentially use that profile to schedule this tool to run on an instance type that is very well suited to run this particular tool. So the cost-aware provisioning, in our experience, we wrote a bunch of papers around it. By understanding the profiles and by using economics, we were able to save around 90% of cost compared to on-demand instances, just by being a little bit smarter about scheduling these tools on Amazon Cloud. Essentially, to kind of summarize, Globus Genomics is a service that makes it easy for researchers to describe, to leverage some of the best practices workflows that are already available. And because the data movement is streamlined because of Globus data transfer capability, it's fairly easy to move the data into Globus Genomics instances, and the resources that are used are provisioned on demand on the Amazon cloud. So let me kind of segue into some of the other capabilities that we're developing as part of the BDDS service, BDDS project, which is a National Institutes of Health Big Data to Knowledge project that we are all funded on. And this is some of the work that we've been jointly working with Steen and Carol's group, is the notion of reproducible research, and what are some of the some of the things that we need to do at an infrastructure level in order for this for data for analysis workflows to be reproducible. And the first challenge is to come up with portable data formats for to make it data easily interchangeable across platforms. And this is where the BD bag notion comes into picture. A BD bag is essentially a sort of a research object that has data laid out in a format that is standardized by the Bagot specification. Bagot is a specification out of W3C adopted by the Library of Congress and others to make it easy to manage digital objects. So we extended that that specification to make it easy to manage big data. So BD bag is a construct that allows that packages data in a format that that allows data to be exchanged between multiple analysis services, and it has tags to describe the metadata and checksum so that you could validate the data hasn't been changed since you last used it. And I'll describe how this all kind of came, how this all kind of makes sense and comes together for reproducible research and doing science at large scale. And in addition to creating tools to package data in a portable format, we also worked on creating unique identifiers for to identify data objects, and this work is called the minimal identifiers. Every data item that that we create in Globus Genomics automatically gets assigned a minimal identifier. So that you can reference it, you can share it, and you can easily resolve it. I mean, one of the challenges of doing reproducible research or doing science at large scale is that all of these tools, the portable data formats, the minimal identifiers, all have to work seamlessly with the analysis tools so that the researcher is not spending time on doing them. So the trick is to make all of this work seamlessly and automatically so researchers end up doing the right thing without having to spend their own time to do it. So a minimal identifier is essentially a lightweight service that works on top of existing digital object identifiers. We've been using something called ARC identifier, which is run out of California Digital Library Initiative. And the min IDs are essentially resolvable. As you can see, you can go to your browser and click and put the identifier there. It will tell you who created it and when they created it and where you can access it and what is the sort of the data that is available in this bag. And we have worked on if the bag becomes a pointer to a large-scale genomics data set, then you could use the min ID would be resolving to a gridftp URL, so you can transfer the data easily using Globus large-scale data management service. So we created a lot of tools to make it easy for researchers to generate identifiers, and we have used these tools in the spirit of eating our own dog food. We have integrated a lot of these capabilities into Globus Genomics so that researchers, when they use Globus Genomics, unique identifiers get created for data sets that are created using the service. So this slide didn't come out very well, but my goal in this slide is to describe a science use case where we have leveraged all of the capabilities that I talked about to do a large-scale scientific experiment. This experiment involves generating transcriptomic regulatory networks, essentially figuring out the gene expression values using machine learning and data that is available in the ENCODE project. So the idea is to create this pipeline that would make it easy for researchers to query, encode, get all the data sets, all the tissue cell lines that correspond to a particular disease type, and do large-scale machine learning and analysis on it, generate these transcriptomic regulatory networks, and publish them to a repository so that others can take advantage of them. So we have used the BD bags that I talked about to generate these data sets. The results from the ENCODE are encapsulated in BD bags. We created a batch execution in Globus Genomics that takes these BD bags. So for example, your query to ENCODE could result in a bag of FASTQ files corresponding to lymphoblast or brain tissue. And Globus Genomics would know that this one unit needs to be analyzed in a batch mode so that a bag could be taken as an input to a workflow and executed in sort of in a batch mode, and provide identifiers for all the data sets that get generated so that you have strong provenance. And then we started using Elastic File System in Amazon to scale up the data that we are running, and put all the results in S3 buckets so that researchers can have the identifier of the results would resolve to an S3 bucket. And also we started looking at encapsulating the tools that we're using in Globus Genomics into Docker containers. So our idea was to sort of have one container description for each tool. So we have over 1,500 containers. We instrumented them with cAdvisor, which is a tool that came out of Google that provides a sort of a very nice profile of the container in terms of how much memory, how much disk has been used. We were able to generate these dashboards and store these profiles in a database that could be leveraged by the profiling service. So we've done this for the transcriptomic regulatory networks pipeline that I talked about before. And the slide, you cannot see it, but if you zoom into if you look at the thing that I'm pointing, that's a minimal identifier, the min ID that we generated for the results while analyzing. This is a lymphoblast cell line data that we have analyzed at scale on Amazon, and all the results have this unique identifiers that are generated. So Globus Genomics, at a glance, it's a scalable service. We have a lot of users using this capability, running thousands of genomes and millions of core hours, and we have reference genomes for over 100 species. Anyway, so it's a very well used service, and led to a lot of great publications for researchers. We created a lot of novel pipelines, great analysis tools that are leveraged by researchers. And this is some set of users that currently leverage Globus Genomics in their institution. More information about Globus Genomics itself can be found here. And information Globus is here, and we're thankful for support by a lot of funding agencies, Amazon, NIH under the Big Data to Knowledge proposal, and the DOE, Oregon, and the University of Chicago. It's a team that I work with that have made all of this work: Dina, Alex, Segan, Paul, and Ian. And that's pretty much what I had, and I think I hope I left enough time for questions. So thank you.
Thank you, Ravi. I will take over now because okay, you're already showing this slide. So to everyone, yes, can you keep the slide? Yeah. So to everyone, you can use the questions function in the GoToWebinar control panel as shown on the slide, and you can post your questions there. Alternatively, even later, you can ask more questions in our forums at ask.bioexcel.eu. We have a special topic there. There was already a question about the slides in the video. After the webinar, later today or tomorrow, we'll upload the recording and the slides so you can review them. So there was a question about, so Ravi, have you had any experience of using workflows that span both cloud and traditional HPC machines?
Sorry, what was the question?
Have you had any experience of using workflows simultaneously on cloud and traditional HPC clusters?
Not simultaneously, but separately. So at Argonne, we have supercomputers, and I work in other scientific domains like cosmology and climate modeling. So we haven't really burst out to the cloud as it may, because the challenge in that is that making data available in where that compute becomes very tedious and takes a lot of time. So we have Globus Genomics running on the cloud, and we have Galaxy for other scientific domains working in traditional HPC at Argonne and University of Chicago, but we haven't combined them both.
Mm-hmm.
I know the Galaxy guys have ways that they have created capabilities to make it work on cloud in a bursty mode, but we haven't used it.
Mm-hmm. What is the largest scale calculation that being done with?
So we have. So one of the collaboration with Emory University here in the US, we have analyzed 800 whole genomes simultaneously. It took like 5 days to analyze all of them, and that was a lot of compute. We were launching hundreds of instances in parallel. So that was one of the biggest thing that we've done, and this ENCODE is also turning out to be a large-scale analysis activity. So we are planning to analyze all of the ENCODE data, which is around 23 terabytes of data. So that's the largest that we've done.
Mm-hmm. Thank you. We have a question by Abdul Rahman. I will unmute him. Let's see if we can hear him. Can you hear us, Abdul Rahman? Can you say something?
Yes.
Okay, we can hear you too. So you are welcome to ask the question directly.
Okay, can you hear me?
Yes.
Yes, probably I've met you, Rafi, in GCC. So the question here is if we want to try the Galaxy on the top of HT Condor with the Galaxy Genomics version with this a lot of tools, it's about 1,500 tools. Do you have an Amazon EC2 AMI so that we can launch our own cluster and try it out, like something similar to the Cloudman?
It's
Galaxy Cloudman.
We don't have an AMI. We have chef recipes that take that. I mean, the chef recipe just takes a stock Ubuntu AMI and runs and configures it. The challenge with the chef recipe is that it has all of the account information and the data bags that we use to launch this embedded into the chef recipe. So yeah, unfortunately, we don't have an AMI that we can share. But if there are any tools,
what you have is something like Ansible playbook or something to generate the whole thing.
Right. Except that it's a chef. Ansible is very similar to chef, but yeah. we do have like an Ansible playbook kind of a thing.
Okay, do you have this somewhere in GitHub so that we can try it out following these steps? Or it
the chef recipe itself?
yes. Do you have a public version of it that people can try it out and?
Right. So that has we don't have it in a public repository, mostly because there are account information sort of hard coded in the chef recipes everywhere. Chef was a very bad choice on our part because we used it because we didn't know anything else at the time. There are other ways of safeguarding these things now, but we haven't really used other configuration management tools. But I'll tell you one thing though. So we are in the process of getting rid of Chef, mostly because we don't need it anymore. We are moving towards using containers. So we have so our deployment process has become very, very simple. We are now, we have a container, a Docker container that the same container that Galaxy guys Beyond has created. We've been managing, we were able to use that within our setup, and that made it deployment on Amazon made it that container made it very simple. So we are moving away from the chef recipe and getting into just using something that Amazon has for managing configuration called CloudFormation template. So that makes it much more simpler for us in managing deployment.
So you have the Galaxy Docker in a container, but you connect this to an htcondor cluster, or this okay, an external htcondor.
Because the container already has htcondor support.
Yeah, I know, because I have made this container with Bjorn, but did you manage to connect this Galaxy container to an external htcondor cluster, because maybe you need to have compute instances separate, not one instance?
In our case, we are all the htcondor we launch is in the context of Amazon, so it just works for us.
I see, because I'm very interested in knowing how did you configure the Galaxy container to connect to an external htcondor cluster. I mean, external nodes. This is very interesting to know.
I mean, I guess I guess we can probably take it offline, but I don't fully understand what you mean by external. I mean, this is a Condor cluster that Galaxy launches, right? It's not external to Galaxy. It's the same Condor cluster that.
So the whole thing inside the container, you mean?
That's right.
I see.
Could I ask a question, Ravi? This seems as an audience, I can't ask questions myself in it. All these tools you have added, how are they described? Are they just normal Galaxy tools in a way, in the end, or where do they live in a way?
They are normal Galaxy tools. So some of the tools are tools that are that an individual research group has developed or used. So they are sort of proprietary to that group, I guess, and they are available in GitHub or outside for others to use. We just created Galaxy wrappers and generated profiles for these tools, so we can run them at scale on Amazon.
And those wrappers, are they available somewhere that we can reuse separately?
Not all of them. Some of them are, but I mean, if there's anything that we tried to set up a tool shed that had all of these wrappers, and the amount of, I mean, to some extent, to get that and maintain it and manage it right has kind of become a little bit of a resource hog for us. So right now, we, if people wanted a tool wrapper, we just point them to our repository so they can download it.
Yeah, I think that's where there's a big overlap with what we want to achieve. in BioExcel with describing tools, and also Galaxy project itself, and also the common workflow language and the bio tools. I guess there could be some room for some kind of collaboration sphere where we can figure out how to do this in a more collaborative way.
We're also thinking, I mean, we also put our Docker containers in this repository called DocStore that the common workflow language people developed. So you could go take a look at DocStore, and that has some of our containers that we developed.
Thank you. Watson, did we have any more questions?
Well, there was another question, but it was mostly answered in the previous question by Abdul Rahman. It was about what would be a good starting point for a newcomer who would like to test these workflows. I guess would be to use these containers that you just described.
Yeah.
Right.
I mean, yeah, we could also, you know, if you contact us, we could create a test account so you can, you know, you can kick the tires or play with it. But the one challenge is, you know, because it's using real Amazon resources, we tend to keep it to a smaller level. Otherwise, we would rake up a huge Amazon bill.
Yeah.
So how is that meant to? How does that work for the current users? Do they have to use their own Amazon credentials, or?
That's right.
Yeah.
So we launched this the whole service in an Amazon account owned by the institution that we are working with.
Is it only Amazon, or is there other kind of cloud backends that we're looking at?
Just Amazon. I mean the challenge is everything comes with a cost, right?
Yeah.
I mean, so our goal is to create a sustainable service, and my goal is to keep the team sustainable. Is to, you know, have a very small number of moving parts and sort of,
get it pragmatic and working rather than a generic
That's right.
try to do everything. Yeah.
That's right. That's right.
Thank you all. We are almost at the hour, and it's time to wrap up the webinar. I would like to remind everybody that if you have other questions that you come up with, you can visit our forums and ask them directly there, and we'll be happy to answer them. And thank you, Ravi, for the very nice talk, and we're looking forward to collaborations in future again.
Thank you for the opportunity. Bye.
Yeah, it was our pleasure, and have a good day. Bye.
Bye.
Thank you.